Hey everybody, this is the last one of module one, and this is tools. A lot of this is going to be memory, so some of it's going to be new. The microscope's definitely new. We're going to do a whole lab on a microscope. We're going to have fun with it, and we're not only going to do one lab with a microscope, but when we get the cells again, we're going to look at stuff under the microscope, and we get after the first year, we're going to use the microscope. So really neat. Everybody loves to use the microscope. I love to use the microscope. I think it's just really cool. I think we get to be Hoke and get to look at things underneath there that we don't always get to see and enjoy. So let's get started. So the tools of a biologist is a lot of the same tools that other scientists use. Uh, so we talk about this beginning part. It's all the same. Just like scientific method is the same. No, well, it should be the same for everybody who uses science. But some people try to manipulate the scientific method, and we know that. We've seen that. We won't go into any of that, but it should be the same. And so for us, scientific method was the same. All of that's the same, and so some of those beginning parts of the tools are the same. And so the first part we're going to talk about is the common measurement, and this is the International System of Units, and that's metrics. Okay, so one of the cool things that I like a lot. I teach biology because I also taught physical science, and in physical science we had to convert from inches to meters and all that kind of stuff. Well, guess what? Whatever we measure in biology is what we're going to keep it in. We're not going to go back and forth a lot of times. We may have to go back and forth from a gram to a milligram, but that's easy to do. Or from a milliliter to a liter, or to a cc, to a centimeters squared, or cc, well, centimeters cubed. But anyway, it's easy to do. So we won't have to deal with those kind of things. Really, energy wise, we'll deal with the joule, but we're not going to convert stuff back and forth. We're just going to talk about this type of food produces X amount of energy or whatever. We usually don't deal with Kelvin temperature because, hey, that's absolute zero, and nothing really lives at absolute zero. Okay, FYI, the closest they've gotten to absolute zero, I think, is like a 4 degrees Kelvin, 3 degrees, something like that. And one of the cool things about that, just a little sidebar, if you don't remember from physical science, is that one of the reasons why they're doing that is electrons will flow very freely and they won't lose a whole lot of energy due to heat. So when they're moving, that energy pretty much is all going in. You're going to get that, recoup all that, most all that energy. You're not going to lose some of it as heat, the entropy part. So, and that's physical science. We're not going to really deal with electrical. We're going to talk about electrical charges, but we're not going to talk about coulombs. Okay, we're going to talk about positive and negatives. We're going to talk about that. Definitely we get to photosynthesis and respiration, but these are things we're not, pretty much Newton's, not dealing with. Okay, but we are going to deal with seconds, liters, meters, grams, those kind of things. And we're going to measure them in that we're not going to convert from English to metric at all. And we're not going to test you on that. So those are all good news kind of things. Okay, but we do need to talk about the common measurements. Okay. Again, we know that these are all based on tens, which I love the metric system. It's a very easy system, to use. We all trying to go to the metric system in America, and it didn't happen. Okay, a lot of very different reasons that it didn't happen. So you can see here, and again, we're not going to get too much beyond. Well, we're probably going to get into the nano world with electron microscopes. Um, Not going to get into any much of that part as far as life goes. Nanos are about as maybe pretty much micro. Micro is about the smallest we're going to go. As far as the biggest we're going to go is probably kilo. And again, we talk about kilograms, but most of the time we're talking about milligrams or just grams. We can talk about kiloliters and those kind of things, but we're not getting way up into here. And we really don't use hecto or deca either. We use deci, centi, milli, micro. So again, we're in this basic. So we're looking at the range. We're in this kind of basic. I didn't make a good one. Basic range right here. Okay, and again, a question mark for the nano guys. They're really, really small. But we pretty much deal with with this group. And again, like I said, Deca, pretty much we don't talk about, or Hecto we don't talk much about. Okay, so again, this should be some things we already know. Okay, so this shouldn't be anything new to us. This might be a little new to us because we're gonna have to do lots of graphs, lots of charts, lots of tables. So this is, but again, one of the cool things is we got a program called Excel, and if we put our independent variable in the right place and our dependent variable in the right place, and we label it right, we're going to get a pretty cool graph. It'll graph it for us. Okay. And if it doesn't look right, then we know that we got our independent variables and our dependent variables mixed. And we'll deal with that. We'll talk about that as we go through and have to graph things and make tables of things that we data that we collect. Okay. So to think about this part, it says you place matter before life and you decide that matter has existed for all eternity. How do you know that the incessant progress of science will not compel science to consider that life has existed during eternity and not matter? And that's Louis Pasteur. So this is something to think about. This is what his thoughts were. Pasteur, again, we've already seen him. He's a chemist. He got rid of the whole idea of spontaneous generation, abiogenesis. He knew life came from life. That matter does exist. That life existed. Again, we go back to Genesis 1 that we were talking about. Matter doesn't just poof bang happen. Life. God created everything. So in a sense, it did poof bang happen. He said it. He spoke it into existence. But when we talk about life, it had to come from life. And again, God is life, and He gave us life. Okay, and so just something to contemplate on page 25 if you want to contemplate some things. So our microscopes. This is a microscope that we're going to deal with. We actually have microscopes that all have illuminators. We don't have microscopes that have a mirror on the end to reflect the light. For the most part, our microscopes have one eyepiece. I don't think we have any that have double binocular eyepieces. We'll talk about all the different parts. You don't have to memorize all the parts right now. We're gonna deal with that in lab. We're gonna look at things underneath the microscope. We're gonna look at how things change, the depth of field, the space. Anyway, it'll make a lot more sense when we do it in class. And so you can start memorizing some of this stuff, or you can just wait till we look at and use the microscope. The big thing is this is using light to shine through an object. And again, so if it's using light to shine through an object, this specimen has to be really thin. Okay, and so you can't have something really thick and kind of see all the way through it. And it's using light. So we can, under light microscopes, we can see a lot of critters that are alive. The next microscopes that we're going to look at, they might be alive when they're put under, but they're not alive when they're done. When we start looking at microscopic critters, pond water and stuff, we're going to put the pond water back because those critters are alive. Okay, and again, that's why you probably want to wash your hands. Probably not want to eat and drink any of that. Okay. So there are two types of electron microscopes. This is a transmission electron microscope. So it emits a beam of electrons through a thinly sliced specimen. A lot of this is right here. I think this is a tobacco mosaic virus. It's a bacteriophage. No, it's a bacteriophage. It's not the mosaic virus. It's a bacteriophage. Anyway, the way that it looks, this is it. So when we use a transmission electron microscope, this is the basic image because it's going through the specimen. So we only get an image of the specimen, and you can kind of see it. They get real hot, so you have to have a condenser. You gotta have something that keeps it cool. Okay, because we're talking about electrons here. So that's the transmission. The other one is the scanning electron microscope, and so that passes a beam of electrons over a surface of specimen. So really, this one gives us a 3D image. So it's really cool looking. It gives us, it'll hook it. Most all of these are hooked to a computer. because you can't look there and draw it. Now, like underneath a compound light microscope, you're going to look through and you're going to draw stuff. Now, you can take a picture, and most of the time, all microscopes nowadays are hooked to a computer. They're hooked to something that takes a picture. So you really, most scientists don't draw anymore. They just take a picture of it. Okay. We want to practice taking drawing. So if you want to take a picture and then draw it later, I'm good with that. But we still want to draw things inside your notebook. Uh, Electron microscopes, you don't have that ability because it's not going to last very long. So it takes a picture of it. And same thing with the scanning electron microscope does the same thing. There's one called a scanning tunneling electron microscope, and that's the one that they looked at atoms with, so they can actually see the atom. Now, can they see protons, electrons, and neutrons? Definitely can't see electrons. Okay, so but they can see the atom itself. So that's pretty quick. That's probably the shortest one. So let's talk about the on your own questions. And it says, why is it important that scientists use the SI units of measurement? We all want to be on the same page. Again, when we report stuff, we want to make sure that, that we're on the same page, going the right direction, that they can repeat our data or look at our data and repeat our experiment. Okay, that's what we want. The difference between light microscope and electron microscopes: a light microscope uses light, electron microscope uses electrons. Electron microscopes, the critters don't stay around long. They get fried. Light microscope, they can stay around a long time, and you can watch them and observe them and eat. And we're going to see that. We're going to see some really, really cool critters. Okay. It says a biologist is studying viruses which are smaller than cells. Which type of microscope would a biologist use if she wanted to study the internal structure of a virus? Well, probably the scanning electron microscope is probably the best. Well, probably both of them. It actually depends on what university you go to and what you have when you're studying that. But pretty much most universities all have both of the electron microscopes now. Western had transmission electron microscope I got to see and use, and again, use it meaning put the stuff underneath there. It's a process also to prep critters to put underneath that microscope. So where it's a lot easier to prep critters to put them underneath a light microscope. So it's all interesting. It's all cool. I think Western does now have both types of microscopes. It's just really, really cool. So if you get a chance to use them with them. So any questions that you got about Module 1, just let me know. We are going to do the experiment 1.1, which is the microscope. We are also going to do the experiment where you guys are supposed to be collecting data about counting the fogs in August. We're going to do, it's not an experiment, we're going to collect some data. We're going to make a hypothesis. And then as we collect the data, we're going to see if this data rings true. If you don't know, the old timers count fogs in August because that's supposed to represent the number of snows that you have in winter. I used to do this all the time with my classes. My students always knew that I had my weather stuff on the side of the wall. They always would come and ask me, say, "Hey, is it going to snow today? Is it going to rain today? What's it doing?" And I would always say, "Okay, look at our barometer. What's the barometer doing? Is it going up, going down? What's the temperature doing? What's the humidity doing?" And a lot of times I would ask them, "Did you remember the dew point from this morning? Did you listen to what the dew point was?" Because all of that plays into what's gonna happen. We have to have a good dew point and a nice cool temperature to get frost. You can have dew, but you don't necessarily have frost. And so it's just really cool stuff. We would do this, and I will tell you when we start taking this data. Previous data that I have, and so we can make a hypothesis of what we think is going to happen. So it's going to be cool with that. And if you guys, the next part of this one was safety in biology. We already talked about safety and being safe a little bit. So again, we're not going to eat or drink in biology. We're going to do it last, so that it doesn't stink up our room. There are going to be a couple of times we're actually going to do stuff during, we'll probably do it before we go to lunch so we can see it after lunch, what's happened. And in those, it's not going to smell, not going to cause any problems. And we're going to need that lunchtime in order for that stuff to take effect, to do something. And then we're going to kind of look at it after lunch. So sometimes we will do some things like that to kind of break stuff up. But for the most part, dissection, looking at critters, all be in the afternoon because it's going to be things that might have an odor. So we don't want that around in our classroom all day long. And we're going to say that, yeah, we're going to wash our hands again after lab, before lab, depending on what we're doing. Um, we are hopefully going to have common sense in all of this. When in doubt, don't. Just don't. I'm going to say too, you may think it's right, but that doesn't mean it's right. It may be that you can drink this, but should you drink it? Okay, those are all things that we need to ask questions. And again, when in doubt, we're going to err on the side of safety. We're not going to do it. So again, when in doubt, don't. Just don't do it because we don't want people hurt. And again, I don't think any of our labs are going to make that happen, but it could. And I'll tell you plenty of examples during class of things that happened that we were prepared for, but we have safety measures in place, and sometimes our safety measures don't. Stuff's going to happen anyway. Things are going to catch fire anyway. You're going to read something wrong, and stuff's going to happen. You're cutting up critters, and you got gloves on, and you try not to cut yourself, but sometimes it happens. Things happen, and so we want to be prepared for that. Okay. Also, in this chapter, the last part of Salt and Light was about Anton van Leeuwenhoek, so check him out. And then you guys have the study guide for Module 1, which is on page 35 and 36. So if you have any questions about those, just feel free to email me, text me, or ask me in class. We are going to take a look at that and those. And so think about that. So, and if you think about it, you can tell me in class if we actually need a whole video of us me going over these things with you. So, and hopefully in study hall, you can come to study hall and we can go over this there. So again, hope you like it. This one was a lot shorter than all the other ones. This is really what I wanted to try. This is what chunking's all about. Chunking information, small bits and pieces. So enjoy.